So it's a real pleasure and privilege to be with you guys today. As I was thinking about what to talk about, I decided to focus sort of on three areas where I feel we've made some contributions, but where there's still a ton of work to be done. And so as you work out your interest in this area, for me, the number one message is that there are plenty of really, really important and open questions in the field. And then secondly, that algorithms actually make a huge difference in a lot of what I'll be talking about. And I'll try to, kind of really drive that point home. It often feels, for those of us that come in from an analysis side, that when you start getting involved in biomedical research data analysis, a lot is driven by the experimentalists or by the big programs. And so you think, ultimately, what's the contribution that an analyst can make? And it turns out to be actually quite important. A lot of decisions and then ultimately policies are going to depend on the methods and systems that we bring to bear. So I'm going to start with this photo. How many of you saw the State of the Union address last year? Not this year, but the year before that. All right, so you may have seen President Obama talk about his precision medicine initiative. And to say that the president views this as a top responsibility is actually understating it. We had a discussion with the head of the Office of Science, Technology, and Policy, and they came down and happened to be at a talk of where the director was, and he said, the president wakes up and thinks about three things: ISIS, the economy, and the precision medicine initiative. So don't fuck it up. I mean, those were literally the words from the head of the Office of Science, Technology, and Policy. And in fact, Obama just published last week in JAMA a sole author paper on sort of his vision for research. So it's actually pretty amazing to have the President of the United States fundamentally interested in a problem that many of us in this room may end up contributing throughout our career to. And if you think about PMI, it's actually an idea that has been pushed by many different groups, and I give Francis Collins a lot of credit. I mean, Francis has been kind of like a dog on a bone about building a 1 million-person cohort, and he's really sort of seen this administration as a partner in making that happen. And so why? What's the goal of bringing together millions of potentially sequenced human genomes with electronic health records? And at a very high level, I would say that the reason we're investing billions of dollars as a country and a community in this is really kind of best exemplified by this story. This is the PCSK9 story. How many of you have ever heard of PCSK9? All right, cool. Well, so this is why there's so much interest in this. So PCSK9 is a very interesting gene. It's a gene that was implicated in the first rounds of genome-wide association studies with increased levels of LDL, or so-called bad cholesterol. So there are common variants in and around PCSK9, and the evidence is pretty strong that these have a modest impact on your levels of LDL cholesterol. And in fact, the whole notion of Mendelian randomization has started with these kinds of LDL genes, where you can look at genotypes at the LDL GWAS loci, and they're actually predictive of clinical outcomes in cardiovascular disease. Okay, so there was a lot of interest in this gene, and then Helen Hobbs and Jonathan Cohen did a beautiful experiment where they looked at families that had naturally low levels of LDL, so naturally low levels of your so-called bad cholesterol, and they resequenced this gene. And what they found, it was a multi-ethnic cohort of African Americans and white subjects from Dallas, that people with knockouts in this gene were overrepresented in that group with naturally low levels of LDL. Okay, so if you have one loss of function mutation in this gene, your average bad cholesterol is something like 79 to 90 deciliters per ml per kilogram or whatever it gets measured in. And that's naturally a really, really good level, right? That means you can eat as many hamburgers as you want and your bad cholesterol doesn't go up. And in fact, they found one woman who was a compound heterozygote for 2 different loss of function mutations. She was an aerobics instructor and had an LDL of like 25, right? Doctors didn't even know you could have an LDL that low. So what's happened since then? Well, 5 different companies have created monoclonal antibody therapies against this gene, and they've been able to demonstrate that it's a sort of next generation of statins that's lowering your bad cholesterol. And what's beautiful about this is that genetics gives you further evidence that lowering LDL is actually causal in the pathway that leads to coronary artery disease, and with the evidence that these people that have knockouts have basically been dosing since they were born with this, it's safe and effective in reducing this biomarker. The flip side of the story is that HDL, so-called good cholesterol, the markers that predict your good cholesterol, on the other hand, have no impact on whether or not you will have a heart attack. And in fact, 5 different drugs failed in clinical trials precisely because they raised your HDL but had no clinical impact. Okay, so if you're now the head of drug development and you're looking at this data and you've got $500 million to invest, are you going to put them in HDL drugs or in LDL drugs? Right? Well, clearly LDL is far more promising. And one of the reasons that medicines are so expensive in the United States is that many of the compounds that we try to put through trials end up failing somewhere along the path. So 90% of compounds never end up making it to an actual phase 3 trial. But the ones where the evidence is coming from genetics have a far better success rate. So that's really one of the main drivers, in fact, that drug companies have begun to invest heavily in this area. We need far better EHR data linked to genetics in order to push that program forward. And some of you may have heard about a drug company called Regeneron that's partnered with Geisinger Healthcare System. They've already sequenced 200,000 exomes in their community in the Geisinger healthcare system, chosen with these extreme phenotypes. So this is great. I mean, it could really be a wonderful shortcut into important pathways that we could punch therapeutically. Another area where, of course, this is being seen a lot is in cancer treatment. So there's a big drive, and the first part of PMI is about a $70 million investment in cancer, where they're going to be sequencing tumors and normals and trying to come up with recommendations. So, that all sounds wonderful. So where's the problem? Okay, so one of the arguments that we and others have made is that because the vast majority of these studies have only been done in populations of European descent, and we had a paper that shows one particularly egregious statistic. So back in 2011 when we published this, if you just asked what proportion of participants in these kinds of large-scale studies are of European descent, something like 96% of the participants were of European descent. Okay, so when you do studies in other populations, you tend to use smaller samples. They tend to be underpowered. So really, a lot of what we think we know is only coming from one corner of genetic diversity space. And so it's logical to ask, is this a problem? Maybe this makes sense as a place to start, but what would we be missing out? And so I'll give you kind of a couple of areas where I think this is going to be really important and try to talk about where there are opportunities for algorithmic improvements. So the first part of the talk is really going to deal with the involvement we've had in large consortia to help drive the development of community resources and reagents for multi and transethnic genetic mapping. So we're part of a consortium called PAGE, which has now designed and genotyped a multi-ethnic array in 50,000 people. It's the largest US minority cohort, mostly Hispanics and African Americans. We're also part of the large-scale sequencing program, which is going to do about 200,000 exomes, as well as ClinGen, which is a community resource for annotation of clinical genomes, which I'll talk about in the second part of the talk. Okay. And then lastly, we'll talk about data security and data sharing, where we need to give some thought to how we expose this data because using just very simple population genetic first principles, you can demonstrate that there are vulnerabilities in the theoretical design of how we're thinking about sharing all of this data. Okay, so some of the driving questions in this work include: what are the arguments for studying diverse populations in a clinical genome research setting? What do we hope to benefit? What are the potential scientific gains? Secondly, once we've identified genes that are clinically relevant, right? Once we move and establish that the CFTR locus is implicated in cystic fibrosis, BRCA1 and 2 are implicated in hereditary cancer, P10 in another set of hereditary cancers, and so on. How do we go in and really understand the allelic series that is relevant for public health in these at these genes and across human populations, and give guidance to community and public health setting where you've got to make decisions about what to genotype, whom to genotype, when to genotype, how to use that information if you really want to move the needle and begin to bring genomics into healthcare. And then we'll also talk about the problem of EMR decision support. It turns out that last mile is a huge hurdle in rubber meets the road kind of decision making. We could have all the most wonderful studies we want, but if clinicians aren't using that information in routine care, then it really is sort of a loss on our side. Okay, so as I mentioned, several years ago we had this paper in Nature, this commentary that argued that from population genetic first principles, as we sequence more and more genomes and begin to query rare variants for association with chronic complex disease, then the design of those studies are going to become far more important. Right? If we're talking about common genetic variation, variants that have been around in the human gene pool for 50 to 100,000 years, that were present at the out of Africa diaspora, well, a lot of that variation ends up being shared across populations, and because it's common, it contributes to heterozygosity, so it contributes to our most common measures of genomic variation. But if we're thinking at an interpret the genome level, there's going to be a huge impact that investment in European populations is going to have in our interpretability of genomes from non-European populations, and that's one of the points I want to drive home. So here's one of my favorite examples. This is a genetic variant in the HLA system. It's called the B*5701 allele. All of these genetic systems that we've studied for many, many years have canonical alleles that those that work on those systems know, and this happens to be one of them. And the reason that I bring this example up is that there's a kind of very close one-to-one mapping between carrying this allele and a particular adverse drug reaction called abacavir hypersensitivity. So abacavir is a drug that's given to HIV-positive patients as part of the triple cocktail. And what's interesting from a population genetics point of view is that there's huge variation on a global scale in the frequency of this variant. And if you happen to be HIV positive and carry this HLA allele and you get prescribed abacavir, within a couple weeks you're going to have a very bad reaction. You get this horrible skin rash, you get very, very sick. Your body, basically mounts an autoimmune response, and this is an immunocompromised host. And then the second time you get prescribed, if you were to be that unlucky, you're going to die. Okay, you're likely die. It's such a bad reaction that you mount that it's really just a terrible decision to give abacavir if you carry this H allele. And the ironic thing is, this is a test that costs pennies to figure out. There's just a couple of markers that you need to genotype. So when you look at publicly available data on the distribution of this variant, it's at highest frequency in the Gujarati. All right, so it's something like 15–20% of the population is walking around with this HLA hypersensitivity allele. Then the second most is in the Maasai. The third most is in the Utah collection, and then it happens to be absent in this particular sample from Tokyo and from the Yorubans. So I don't care how good a population geneticist you are. There's no sort of first principles that would guide your intuition immediately here, right? This is a very empirical question that we just simply need to go out and collect data across different populations on this particular SNP and then guide whether or not you genotype people, right? You don't want to kind of go with first principles here. You actually want to go out and collect the relevant data. And so this is an example of, if you're designing, say, a pharmacogenomics panel for a hospital, then it's really relevant. And you need to understand what to put on there, what you're seeing in terms of your particular patient population, because you don't want to miss this one. I mean, there's no debate in the scientific community about whether or not genotyping this would be a good idea before prescribing, right? Everyone agrees on that. It just doesn't get done. Right, and so you got to think about that in terms of how you want to develop these going forward. So what we've done is really try to build collections that'll give us insights into this, and this includes just a ton of different projects we've run up and down the Americas and West Africa and South Africa, in Europe and other places, because a lot of this data actually is straightforward to collect, particularly if you could then link it back to phenotypes. I'll give you a couple of examples from projects that we've undertaken in this direction that, in our mind, really speak to the importance of this kind of multi and trans-ethnic approach. The first is a project that we had several years ago led by this guy named Sean Miles. So Sean was a postdoc in my lab who was very interested in of all things, blonde hair. Okay, I thought it was kind of an interesting question. And so Sean came in and he showed me this photograph. This is a photo of kids from a place called the Solomon Islands, which is a sovereign nation off the coast of Papua New Guinea. And he said to me, he said, Carlos, I want to work on blonde hair. And I said, Sean, look at this kid's U.S. serviceman's jacket. I could tell you the genetic basis of blonde hair, right? It's a Captain Cook allele. His dad, was a European with blonde hair, and that's widespread in the population. He said, no, I actually think it's likely to have a different genetic basis. So what I'd like to do is go and build a cohort and collect kids, and we can genotype them. I said, John, that's crazy. You're never going to get money to do that. I said, all right. So he went and, like all good postdocs, didn't listen to me. He got a grant, went out and collected 1,000 samples from islands in the Solomon Islands, and then came back. And Sean had a very clever idea, which was just to focus on extremes of the distribution. And to make a long story short, you could read it all in this particular paper. He was able to demonstrate that there was only one region of the human genome that was very strongly associated. Our initial sample size only had a couple hundred kids in it. And we could easily, the minus 10 log p value here is about 10 to the minus 20th. The effect size is 2 standard deviations out in terms of spectrophotometric measurements of hair pigmentation. We could demonstrate that it was in a gene called tyrosinase-related protein 1. Here's the distribution of hair pigmentation for about 1,000 kids. Here are kids that are homozygous. Here are kids that carry one version of the allele. You can clearly see these two distributions are basically indistinguishable. And then the kids that are homozygous for this change not only have, are basically 2 standard deviations out from the distribution, but they're walking around with homozygous cysteine at a highly conserved amino acid residue. As a good PI, I became very interested in this project at this point. And we really spent a lot of time in functionally characterizing this. And it turned out to be a fascinating story because this TRP1 gene had been studied in other systems. So it's the source of this canonical difference in black versus light mouse. In dogs, it's actually been knocked out and segregates in certain breeds. So these two are litter mates, and one is brown and one is black based on a knockout of TRP1. And in humans, actually, there's a knockout of the allele that was described in a family from South Africa, which is one of the first cases of a type of albinism called oculocutaneous albinism type 3, or albinos of dark skin, right? So it's a super interesting gene. It's been studied to death. And so we were quite excited that we were able to find this new allele in the gene that was implicated in blonde hair from Melanesia. What makes it particularly relevant for our discussion, though, is that this is different than the genetic basis of blonde hair in Europeans. So this is what one may call LA, or chemically induced blonde hair variation. But if you have naturally blonde hair and aren't Melanesian, then you would tend to have a mutation in the OCA2 KIT ligand region. You don't have this TRP1 allele. Okay? So here's blonde hair, a dead easy phenotype to study. There are two major human populations that have blonde hair, Melanesians and Europeans. And when you look at the second population, you realize that they have blonde hair for a totally different reason. Okay? So why would we presuppose that this is not likely to be true for lots of other traits that we're interested in? Diabetes, right? Why would we think that the genetic basis of diabetes is going to be identical across different human populations? In the Indian subcontinent, diabetes onset is a much lower body mass index than in European populations. Likewise in Japan. Right? So it stands to reason that while many liability alleles may be shared, a fair number of them may be private to populations. And again, population genetic first principles tells us that anything that's down below a couple of a percent is very likely to be population private. So we spent a fair amount of time trying to make this argument, and this is work that builds on Simone Gravel's work in the 1,000 Genomes Project and others, where we did exactly that. We came up with a series of very simple sharing statistics and basically exchangeability of genomes across populations. And early on, we knew this from the pilot phase of the 1,000 Genomes Project. It was pretty clear that for common variants, European populations are basically interchangeable. Even European and Chinese or European and African populations shared a fair amount of the variation at comparable frequencies, if that was common genetic variation. But for rare variation, which we now know is the bulk of variation in the human genome and what you would predict from population genetic first principles, that the modal class would be singletons. Even if you're not talking about singletons, you're just talking about rare variants, things down at a couple of a percent, these tend to be extraordinarily population private. And this is where we're driving genome-wide association studies now, so it's very, very important to think about this in the design. So what we've basically argued to NIH and others is that there's plenty of opportunity to do large-scale genetic association studies in non-European populations because we've probably not found even the modest frequency alleles. And when this has actually happened, similar to when we did our study on blonde hair, there was a very nice study a couple years ago from the Broad where they did a GWAS study in Mexico and found a type 2 diabetes allele that was at 25% frequency in Mexico, absent from most of Europe and Africa, that they calculated explains something like 15 to 20% of the variance in ethnic group specific risk. Okay. Likewise, in a host of different systems, when folks have gone in and actually done multi and transethnic GWAS, you're able to demonstrate that not only are there new alleles at genes you've previously studied, they're new genes that you didn't even know about are relevant to disease, and in fact maybe more relevant in some populations than in others. So in order to really give this some clarity, we've worked with the MEGA consortium to design an array specifically designed for multi and transethnic mapping. This is largely the work of Emer Kenny and Chris. So Emer was a former postdoc of mine who's now at Mount Sinai, and Chris Gnu and Jen Vacek in my lab have worked with her to design this array. We've now been working with Illumina to commercialize it. It's got 1.7 million features on it. I'll walk you through some of the most salient design elements because I think they speak to this issue that algorithms are important. So we wanted this chip to do two things. One, have clinically relevant content on there that we could genotype in as many people as possible. So known alleles either at clinically relevant genes like hereditary cancer genes or at GWAS implicated loci, as well as enable a new generation of genome-wide association studies from a new designed SNP backbone that was specifically engineered for multi and transethnic mapping. So this is sort of the breakdown of the array. Part of it is custom content, a fair amount of it is clinically relevant content, and there are a fair amount of the work that we did on redesigning the scaffold. And the reason that this is quite interesting for us is that you could imagine a couple of different strategies that you could take in thinking about a tagging sort of algorithm. So you could go through for each SNP, calculate how well it imputes other variants, look at this across populations, and then sort of maximize. Alternatively, and then you sort of repeat until you sort of have prioritized the markers from your selection. An alternative strategy, so if you were to do that, it would look something like this. Apologize. Where you would go through and just sort of maximize how well you're mapping regardless of which population you're looking at. Okay, and that's what we're seeing here. You've basically gone through for each marker. You've gone and calculated how well it's tagging within each of these superpopulations. This was from 1,000 genomes. Alternatively, you could begin by looking across populations and see how well you're doing within each of the supergroups in terms of tagging, and then sort of re-rank so that you're preferentially choosing markers that are maximizing tagging ability within the subpopulations. Okay, so the only reason I raise this is that when you've got a finite amount of real estate, as we did in this project, the choice of SNPs is really going to be geared by the choice of problem you're trying to solve. So we, from the beginning, designed the tagging algorithm to maximize within subpopulation tagging, specifically because we wanted to do this multi and transethnic mapping. Does that make sense? Okay, and so that's a unique feature of this array. You can actually go in and demonstrate that for the African and Asian populations, it actually does better than the mega 5 million array, given the set of statistics that we've chosen. The other part of this that for us was very important is to put known pathogenic variant on this array. So we actually went through and worked as part of our ClinGen ClinVar project to choose about 28,000 variants that had different assertions made as to whether they were pathogenic, benign, likely benign, and so on. Because one of the big challenges that we're facing as a community is as we move from clinical genetic testing that is ordered by a physician in a context of a particular diagnosis. Let's say they've looked at a pedigree. It's a pedigree with multiple women in it that had breast cancer before 50. Well, given that information, you have a much stronger prior that this is a family that may have hereditary breast cancer relative to doing ab initio genome sequencing. Okay? And so the posterior on the likelihood that a mutation that you find, say, in BRCA1 and 2 is going to be pathogenic is far higher in the family that had BRCA1 and 2 sequencing done based on a clinical diagnosis of hereditary breast cancer to a random person that walks in the door. Okay? And that information is actually really, really important in making this adjudication as to whether the variant is pathogenic or not, particularly at genes like BRCA1 and 2, when people are going to go out and have double mastectomies based on your adjudication. Okay? And so because there is so much uncertainty into how this process works, we wanted to put on there a fair number of known benign and known pathogenic mutations so that we could see what the determinations that one could make based on frequency, based on prediction algorithms, and so on. And there's a fair amount of really relevant content now on this array. I mean, it sounds kind of crazy to think about doing screening using this array system, given that we've got the ability to do targeted capture and sequencing. But in fact, we've already got hundreds of known BRCA1 and 2 variants on this array alone. Okay, so that's relevant as we want to get frequencies across populations to understand the natural history of these alleles, to be able to compare them to benign alleles that might serve as a null model against which you could test and so on. So we've done this in particular for the genes that are in the ACMG 50 list of incidental findings. So this is a list of 50 genes that the American College of Medical Geneticists say is so important that if your doctor were to find out your genotype at these genes, even if it happened through ancillary testing. So let's say they want to do testing for carrier status screening testing because you and your partner are going in for in vitro fertilization. Just think about that example. So they run you guys up for carrier status, but they happen to find out your BRCA1 and 2 status or your status at Huntington or P10. Then the lab, because of this new regulation, is duty-bound to return that result to the physician, and the physician to interpret that for you, even though they may have never ordered that test. Okay? So here are people going to the clinic, having information about their clinical genetics, ordered up, and then a physician's going to have to interpret this and give this result back to patients. Okay? And the catalogs are actually pretty terrible, particularly in that setting where you've got an incidental finding. So for us, it was very, very important to make sure that we understood the population genetics of this to be able to give some guidance. The other part, and I don't know how many of you know this, but in the ACMG guidelines, how the determination of pathogenicity is made, right? When you get your test sent off to one of these clinical genetic testing companies, Quest or Emory or Harvard or Stanford or whatever, UCLA, they use the population allele frequency data as part of the determination, right? They've made these kind of arbitrary decisions about how to do that with very little input actually from population geneticists. So, and you can actually get very strong evidence that a mutation is pathogenic depending on where this falls out. And I'll share an example in the next section about where this could be really problematic. So for us, it was very important to bring some clarity to this problem. We've worked in particular to try to get data on diverse populations from the Americas, so we at least have some coverage of African Americans and Hispanic Latinos, which are the two largest ethnic minority groups in the United States who are going to be seen through this kind of a system. You could do all kinds of things with these data. I mean, that's what's so exciting about it. It's 50,000 people. I'm showing you the very first instinct thing to do. This is data that we actually started working on less than a month ago. But this is a comparison of our assertions, whether mutation's pathogenic, likely pathogenic, whether there's conflict over how different labs have determined this benign or likely benign or benign, and the total frequency in our page cohort versus the frequency in EXAC, which is the Broad's exome server, right? And right now, basically the de facto source that gets used in clinical determination. And so you can see examples here. I mean, these are the ones for me that are particularly worrying. These are variants that we've asserted, or we as a community have asserted, are likely to be pathogenic or pathogenic. And if you were to look them up in EXAC, you've maybe seen them once or twice. So you would think they're really, really bad. They're very, very low frequency. But when you actually go out and you genotype them in a big multi-ethnic cohort, they reach appreciable frequencies of 1 or 2%, right? So these are very unlikely to be super bad pathogenic mutations unless you've got additional evidence, right? And it's simply from a function of using this as your reference, even though there are 70,000 sequenced exomes here. So it's not a small sample size. It just happens to be a largely European a cohort there. So for us, it's incredibly important to get this right. If people are going to be making life-altering decisions based on these determinations, it's important that we bring the right data to bear on these problems. Just to give you a kind of another visualization of this, this is one of the most important variants out there. This is CYP2C19 variant that has to do with your response to capiligril, which is a blood clotting drug. And you can see this is an example of where, in certain populations, it it reaches 5 to 10% frequency, but in certain others, right, we've also genotyped HGDP on here, particularly in Southeast Asia, you're seeing it in 50, 60% of the population, right? It's incredibly important information to know, particularly if you're testing people out in this part of the world, okay? Here's another example. This is CYP2D6. And this is one that's very controversial, actually. CYP2D6 has to do with your metabolism of coding. You can see that it's actually pretty absent in Eurasia, but if you happen to be of African ancestry or West African ancestry, then it reaches comparable for 25% frequency in some places. And you have no response to coding, which is terrible, right? And so this data has actually been used by most children's hospitals to get rid of coding altogether. Right, so if you go to Miami Children's Hospital, for example, one that I've worked with, they no longer prescribe coding. Why? Because they know something about the CYP2D6 gene. They know 10 to 12% of the population doesn't respond well, so they're like, screw coding altogether. We're going to go deal with other narcotics. Even though it's a very good drug for some small number, for the vast majority of the population, it's an excellent drug. It happens that 12 to 15% of the population, particularly those of African ancestry, don't respond to it. Right? This is a solvable problem. This is not something you're going to need to guess at, but instead of dealing with this, it just totally eradicated what's a perfectly great drug and analgesic to use. Okay? And so our hope is that you can begin to really tailor this in a much larger setting than folks have envisioned before, right? When you think about pharmacogenomics, you think about it getting used in very elite medical centers, but there's no reason you couldn't deploy a $50 array across most community hospitals and significantly reduce the errors in prescription by carefully choosing and putting this information together and putting together a report that could be used by them. The other thing that we've worked on is actually what you call the last mile problem, really putting this into a clinical decision support framework so that clinicians will use this. There's a large software company called SAP. Maybe some of you have seen this when you go to the airport or when you watch basketball. Statistics brought to you by SAP. It's the same. They're the largest software company in Germany. So we've partnered with SAP to bring together electronic health record data, full genome sequence and interpretation data, as well as wearables data, all into a single framework that they can use and make available to hospital systems. So we're partnering this in parts of Germany, as well as practices in the United States that would like to sign on. All right, so what have we learned so far? I would put it into this sort of broad category that, much like the rest of the population genetics community, common variants are actually pretty rare, and rare variants are quite common. And the implications of that in a diverse society such as ours is that if you don't broaden representation in the set of participants that you use in your clinical research, then you're, number one, leaving lots of interesting biology on the table, and number two, potentially doing harm and increasing health disparities, particularly in an area like genomic medicine, where you're going to naturally already have an increase in disparity simply due to access. And so you might find novel variants of previously associated loci, so at both LDLR and PCSK9, these are involved in your metabolism of bad cholesterol, as well as new genes, completely new genes underlying interesting biology. So we've been working closely with NIH in their design and implementation to make sure this happens. And you might have seen that in particular, one of the planks of the Precision Medicine Initiative is that it's not only a participatory cohort, but it's a diverse participatory cohort. And so I think that NIH is getting the message, but it's incredibly important that we also continue to be involved as this gets rolled out, because there are lots of decisions that need to get made. And it turns out if you're not at the table, then the decisions could go other ways. I think challenges ahead, I would say that one big challenge is nobody has really sort of properly thought about design and optimal design. I would say PMI's approach is to just take all comers and sequence them proportionally. That of course means that the individuals that are from rarer ethnic groups are going to be rarer in the data set, and you're going to potentially not have the sample size that you need to be able to make the right assertions. I would say one of the big challenges, and I'll talk about this next, is this these variants of unknown significance. And it turns out that variants of unknown significance are particularly problematic in underrepresented populations. So for BRCA1 and 2, for example, a woman of European ancestry could expect an inconclusive test maybe 2 to 3% of the time, where the analyst looks at it and says, we've never seen this allele, we can't adjudicate it. Whereas if she were Indian Asian, that number is closer to 12 to 15%. Okay? So we view that as quite a problem and something that needs to get addressed. And then, you know, really, for a lot of these, you're ultimately not going to be able to adjudicate until you do the right functional test. And I want to talk a little bit about that, because this isn't just going to get solved algorithmically. Okay, so the second part of the talk has to do with this problem of pathogenicity prediction in clinical genomics, and one that is closely tied to the first part of the talk. And to motivate this, I want to tell you, I don't know how many of you saw this piece. It was in the Atlantic about a year ago from Ed Young. If you haven't, I encourage you to read it if you're interested in this area. So this is an example of what can happen in this kind of clinical genetic testing. It follows my colleague Heidi Rem, who runs the clinical diagnostics lab at Harvard. And it's a particular case that she had where this family came in for carrier status analysis, and they did some cell-free DNA sequencing of the child in utero. And when they looked at the data, it suggested that there might have been a quite terrible pathogenic mutation in a gene that would lead to a whole host of phenotypes, including mental retardation in a child that the family would basically need to take care of most of its life. However, it was still a new allele. Nobody knew exactly how to interpret it. And so the family basically went home with a variant of unknown significance and not knowing exactly how they were going to deal with this. Heidi kept working on the problem, got kind of a bee in her bonnet about this, and then it turned out that about a week later she found that that allele was actually pretty common in Turkey, where this family happened to be from. It had maybe 14–15% frequency in Turkey. The Mendelian disorder is very rare, and so it's unlikely that that allele is not causing a Mendelian disorder. Right? Does that make sense? You can't have something that happens once in a million if the allele's at 20% of the population. So she went, she contacted the family, and the family said, okay, well, it would have been good to know that before we terminated the pregnancy. Okay? So, it's one example of, how this happens, and, we should be able to do better, right? And she's been kind of haunted by that case, and that's the purpose of that. I encourage you to read it. It's a very interesting article. So what are we doing about it? Okay, so we're part of a consortium called the ClinGen Consortium, which is aggregating clinical genetic testing data, and our part of it is to try to bring better algorithms to bear on the problem, and we were particularly interested in trying to use machine learning algorithms to adjudicate pathogenic from non-pathogenic mutations using what one could call a meta-predictor that takes in a series of predictors and additional features of the data that you try to use. And of course, this is, for those of you who've done this kind of work, it's pretty straightforward. You sort of try to take a training data set of known pathogenic mutations, known benign mutations. You have a hyper dimensional space here, and you're just looking for a function that clearly separates based on whatever objective function you choose, the input space. And then through cross-validation, you can see how well you're doing. So, and then ultimately what you want to do is apply this to a much bigger data set that includes many variants of unknown significance and potentially pathogenic and potentially benign mutations, these 2s and 4s, that you can hope somehow improve the adjudication of. So as part of this project, we've built 4 such predictors. One is called REVEL, which is for non-coding variation, and this is a kind of random forest. Another one's called FIRE, which is for non-synonymous, and it's sort of an add-on to CAD. I'm actually not going to talk about them. I'll be talking mostly about this CFTR predictor, which is for cystic fibrosis, because it kind of highlights a lot of the problems in this whole area. These are kind of uber genome-wide predictors. These are gene-specific predictors. And what we've done is now put this all into the curation interface that is being used. I'm going to skip this. This is sort of the design of all of the ClinGen data, which isn't as relevant here. All that's relevant now is that we've now built this curation interface and have aggregated a bunch of these predictors and are serving them up so they can be used in the curation process. But this is great in terms of putting this all in people's hands. It's not great in that the predictors overall just leave a lot to be desired. And so let me just walk you through the CF data so you can get a sense of, a sobering sense of where we really are as a field. So cystic fibrosis is probably one of the most studied Mendelian recessive disorders for a couple of reasons. First is that it was the first Mendelian disorder to be cloned, right? The first gene to get cloned. So this is what made Francis Collins, head of the NIH, famous. He cloned the gene for CFTR and demonstrated that it was the cause of cystic fibrosis. Secondly, it's the most common Mendelian recessive disease in Europeans and European Americans, partly because there's one allele that's at 2 to 3% in most European populations, the so-called delta 508 allele, and so it's part of routine screening. So any of you who've had children or know of these small humans called children, on the day the children are born, they get a blood spot test. This is one of the things that they're testing for. Okay, so it's about as studied as things get, and most people, something like 70% of cases, carry at least one copy of this allele. Okay, now that doesn't mean that everybody who has cystic fibrosis because of delta 508. Okay, and in particular in the state of California, where most of the kids born today aren't of European ancestry, that number is far smaller. And so if you think about this gene, there's a ton of variation in the CFTR gene that you need to worry about, because we need to figure out whether these kids need very intensive follow-through and have cystic fibrosis, are going to need to basically be seen in the clinic by experts, or whether you can clear them as not having cystic fibrosis. Maybe they're cystic fibrosis carriers, but they don't have CF itself. Okay? So what have we done? We've taken and aggregated about 2,000 potentially pathogenic, benign, and variants of unknown significant variants at the CFTR gene. This includes data from a database called CFTR2. There's a fair amount of clinical information associated with it. This is another database that was made specifically to create a CF predictor. We've got data from dbGaP and EXAC. This is basically population-level sequencing. Then we've got data from our CF center at Stanford. So these are kids that we've followed in the clinic, about 600 of them, and we've got good clinical outcomes. Then the last is from the Stanford Molecular Pathology Lab, where we have the contract for the state of California. So every kid with a positive blood spot test, if you think they've got CF, we've sequenced the gene at Stanford. Okay? And this is a huge sort of collection of what the state of California has to deal with in terms of adjudication. This is, in my mind, the scariest figure I'm going to show you all day. Okay? And it's just a simple pie chart of those 2,000 variants and for how many of them we've been able to confidently call them a CF-causing allele, how many, maybe more importantly, we've been able to call as a non-CF causing allele. But the vast majority, 75% of the variants in the most studied gene in the human genome, we still have no clue whether or not they cause cystic fibrosis. Okay? To me, this is a failure, right? We have to be able to do better than that. This is a gene that gets, every kid born in the US gets screened for this. And 75% of the variants that we find, we have no idea what to do with, right? So, on the days that your friends are telling you about the best app to predict your diabetes risk from your genome. Tell them, buddy, we can't even predict whether somebody has cystic fibrosis, and we sequence everybody. So what can we do? And let's think about this problem from an analytical point of view, because the answer should be there, right? This is a simple Mendelian disorder. This is not some very complex trait you're trying to deal with, and we understand a lot about the biochemistry and chemistry of the transmembrane. So what did we do? Well, we did what any half-wit computer science person might do. So you guys got probably much better ideas. We're not particularly good computer scientists. So we just took a bunch of particular predictors, right? Let's make a list of all the things that one could try to predict on, and these might be features based on sequence, features based on conservation, features based on molecular dynamics simulations, and so on. Then the other thing we did is just choose a bunch of potential objective functions, okay? And these are ROC curves. On the left-hand side, you've basically got different predictors: gradient boosting, random forest, support vector machine, GLMs, and so on. And on the right-hand side, you've got different underlying predictors. And we should all be kind of a little bit embarrassed. Maybe I'm particularly bad at running these things, but none of these have an area under the curve greater than 80%, okay? And on a good day, our best predictor, we were able to get it to be like 84%, okay? That means that more than 15% of the time, it's making the wrong call, okay? And this is where you've taken the biggest data we could assemble, the best predictors we could assemble. You try a bunch of different weighing functions. You never break through about 80 to 85%. And Rachel Karchen spent 2 years on this problem, and the same is true for her, okay? And so it just tells you that trying to predict at this level of granularity is probably not the right level, okay? So you're trying to predict whether a kid eventually gets cystic fibrosis or not, 15 years down the road based on their genome on day one, okay? So what can you do? Well, it's not the only traits you could choose. We know we've got a bunch of potential cellular traits. So this is an assay that you can run to look at mean chloride conductance. In blue here, I've got all the cystic fibrosis alleles. In red, I've got the non-cystic fibrosis alleles. And in green, I've got the variants of unknown significance. Well, you can train this by eye, right? Anything, and this is our random forest posterior probability, right? So you can clearly see that these guys that are down here with low mean chloride conductance, you can almost just call them by eye as being likely to be CF causing, right? And the same is true for these variants of unknown significance, right? It's not a super complicated problem, right? Whereas these variants of unknown significance that have high mean chloride conductance and look just like the normals, boy, those are easy to call too, okay? So if we just had this kind of data on all the variants, we would immediately do better than how we're doing now. This is another measure. This is the mean sweat chloride, which happens to be the standard clinical phenotype that you would conduct on a patient, because you can just measure this directly on the kiddos. Doesn't do as well, but it's another source of data. So what we've argued, and again, these details aren't important, that what we really need to do is a scanning mutagenesis experiment where you go through and you engineer all of the possible single nucleotide changes in the CFTR gene, and you measure it in a dish, right? And there's some detail about what the right cell line is, and there's some details about, you use CRISPR, you use lentiviral expression, whatever, right? There are all kinds of details, but they're just details, right? They're not strategy questions. The strategy is all about getting far better endophenotype data, particularly on consistently created cell lines. And so we've been working to do this for the CFTR gene and make the data available to lots of folks who like to train their own predictors on it. Okay, this is what this might look like. So this is a fluorescence image of wild-type cystic fibrosis. There's a time course data here. What you're supposed to see is that there's less green here than there, which means that the ions have gone into the cells. These are cells. And then this is the delta 508, and you're supposed to take that there's not much change here. Everyone kind of see that? Now, in the way your machines would see it, this is the time since you add their reagents for the assay. The point is that the fluorescence intensity drops with wild type. There's enough replication here that you get a sense of the noise. And then here's the delta 508, right? You could have computers eating this all day, no problem, right? This is exactly the kind of data that is very easy to digitize, very easy to collect, and then creates the sort of real functional database that you're going to need to properly train these predictors. Because I don't know about you, if I get my genome sequence, I want predictors above 95 to 99%. I don't throw any 80% predictor bullshit at me, because I'm not going to make any kind of clinical decision based on that. Okay? So conclusions, machine learning algorithms are showing better performance compared to separate predictors, but it's nothing to really brag about. We've been able to get some modest improvements, but really to take this to the next level, we're going to need, even for CFTR, good endophenotype and cellular assay data. The other thing that we're going to try to do, and this is really kind of a call out to the community, we're going to try to create a working group of people interested in these kinds of pathogenicity predictors. We can come up with standards for how training data are shared, standards for how objective function data are shared, and so on, because we think it's really important to bring the people developing these kinds of algorithms together so we could think about what features we'd like from the data, so that as this gets produced and aggregated, we can serve it up to the community. All right, the last problem I wanted to talk about, again, so these are supposed to be good algorithmic problems where we've seen that there's a hell of a lot of headroom here. The last one I want to mention is this data security and data sharing problem. And this is one where we were kind of unwitting participants. We just kind of got involved in this problem as part of the Global Alliance for Genomics and Health. So the Global Alliance in Genomics, how many of you have heard for GA4GH? You guys have heard of this organization? All right, so GA4GH is a group of about 300 people, 300 groups, a group of groups that's trying to come up with standards for sharing genomic data in the context of healthcare, okay? And the particular problem that they started with, the first problem they took on is this so-called beacon problem. And the idea is, imagine you're a clinical geneticist or a genetic counselor. You've sequenced the genome of a kiddo as part of an undiagnosed disease. You find some mutation that you think is interesting, but you want to ask, who the hell else has seen this mutation? So the idea behind the beacon is that you can send out, kind of like a beacon, a request. Has anybody seen allele A at this position in the genome at this chromosome? Does that make sense? Yeah, so you send out that request, and you can ping a collection of genomes that themselves may be collections of genomes, and then you get a yes or no answer. You can send this ping anonymously. Once you get your yes answer, then you can go contact whoever claims to have had this genome so that you guys can connect and maybe share the data. Does that make sense? Okay, what could possibly go wrong? Okay, so what are the alternatives? What's the argument for doing this? First, it's dead easy, right? It's a very simple web service, like 4 lines of code. And it's a very simple query and response that you send out. It's efficient for users interested in getting data. You can send lots of pings out, and lots of people could be listening for your pings. And presumably, because you're only getting a yes/no answer, it's probably a pretty safe thing to do. Or is it? Okay, and that was basically what we set out to answer. Okay, and our claim, and this is really the work of Suyash Shingarpure, who was a postdoc at my lab and is now at 23andMe, is that re-identification using this kind of system is provably easy, and in fact, probably takes far fewer pings than most people realize. And so we kind of set out to just show from, again, very simple population genetic arguments that these beacons leak information, and they do so unintentionally, but put together, you could absolutely re-identify whether somebody was part of a collection of genomes or not. And the issue with these beacons is that they often have associated metadata. Like, this is the McLean beacon for psychiatric patients, right? This is the beacon for kids with autism. This is the beacon, And so not everybody's necessarily on board with you pinging and knowing that information about them just from the beacons where they were found. Okay? So we set this whole thing up as a very simple likelihood ratio test. We're given a genome. You're trying to choose between two hypotheses. The null is that the genome's not in the beacon. The alternative is that it isn't a beacon. All the data are simple bits, ones and zeros that tell you whether or not you saw this allele or didn't see this allele in this particular beacon. And you can think about the problem in this way. Let's imagine each of these are genomes. Red indicates a SNP, and blue here is the beacon composed of these genomes. So you've basically got a 0 where you didn't see that variant, and a 1 where you did. Does that make sense? Right? So it's just sort of the union of all of these genomes. So I can send in a beacon and say, have you seen 1 and 4? And the beacon would respond, yep, I've seen 1 and 4. And that makes sense because this genome's definitely in this beacon. Does that make sense? OK. You might also have false positives. Let's say I sent out a ping that said 2 and 4, and that turns out to be yes, except there was never any actual genome that had 2 and 4. Everyone see that? OK. So this would be a false positive of response to from the beacon. However, it's pretty easy to show that as you add markers, you can easily control this false positive rate. Okay, this false positive rate is really a function simply of the markers that you've pinged. And you can think about a couple of kind of limiting cases. This is a trivial limiting case where every SNP is a singleton. So of course, if every SNP is a singleton, then you'll never have any false positives, right? Because every ping you get back is basically telling you whether or not someone's genome is in there, because every SNP in that genome is unique, right? So that's easy. And then you can think about a mixture where you say P of the time or S of the time it's a singleton, 1 minus S it's not a singleton. And then you can basically show that it's all a function of this 1 minus S. And so if you choose something like chance of producing a singleton is 90% or something, then after about 200 pings, you're well within any kind of noise range that you'd want, right? There's a finite number of people on the planet, and 10 to the 10th is a reasonable one. If you want to be 10 to the 12th, you could do that too. Okay? So it's easy to get at what those numbers should be. What about the power to do re-identification? Okay? So you can write this whole thing down in a likelihood ratio test, and I'm not going to derive it for you. For those of you who are interested, it's derived in the paper. The cool thing is that it's a very dead easy statistic. A sufficient statistic is just the sum of the responses. And what you need to know are two constants that depend on the beacon size, capital N, and the mismatch rate among genomes, which is something about the error rate and the quality. And then this whole thing gets parameterized by a beta distribution that we put in as what the background allele frequency is. But that's really sort of a bells and whistle to this whole thing. So you can easily write down this likelihood ratio test. And you can also write down the power. And again, it's a non-central chi-square that I'm not going to worry about. But you can, those of you who want to check the math can check the math. The point is that it's all pretty straightforward. And what's interesting is that even simple simulations give you some insight. So we simulated 1,000 genomes with about 500,000 markers. We put 200 people in the beacon, 200 people not in the beacon, and then just ran some re-identification attacks. And what you can see here, here's sort of the empiricals from our simulation. The dashed line here is theory with an error rate of about 10 to the minus 6. And to have, if you want to sort of control your false positive rate at 5%, 5,000 SNPs, randomly chosen, not even smartly chosen 5,000 SNPs, is more than enough to give you pretty close to 100% power to figure out whether someone's in the beacon or not. Okay? As a comparison, just to kind of put this in an order of magnitude, 23andMe collects data on like 700,000 markers. Right, so 23, I need less than 1% of your 23andMe data to figure out whether you're in a beacon with 1,000 people in it or not. Okay, so that just gives you a sense of the power of this kind of a system. And it all comes out, I mean, it's kind of like paternity testing, right? In paternity testing, it's not the common alleles that you know, sort of nail the dad, it's the rare alleles, right? You have a couple of rare alleles and you see them in your beacon, and the odds of you not having been the person that contributes them is, you know, gets smaller and smaller and smaller very quickly. Okay. What about relatives? Well, it shouldn't surprise us that we can also find relatives of people who are in beacons. So this is the same plot given, this is the actual individual, this is a first-degree relative, a second-degree relative, and so on. And the point is that, again, you're not talking here about millions of markers, right? You're here in the 20,000 marker range, which is trivial to collect, even from degraded DNA, right? So we can take, you know, a thumbprint and sequence a thumbprint, we'll get 20 to 40,000 SNPs from you, no problem, right? So just think about this, right? This has pretty big implications. And this is just sharing allele presence of absence in a collection, right? The same actually holds for a mixture of individuals. So, you know, if you really want to get creeped out, think about filtering, about sequencing air filters and then having people's genomes from a fingerprint, and you can absolutely match those things up, right? From a mathematical point of view, it's the same problem. All right, we then constructed some real beacons with real genomes. This is from a very small beacon, about 85 people. Although, you know, most beacons are small beacons, right? Most hospitals that have done this kind of routine genome sequencing only have a couple hundred in there. They don't have tens of thousands of people whose genomes have sequenced. With this, it was easy to show that if you've got 100 people in it, you know, with 100 or 200 SNPs, you could easily figure out whether they're in that collection or not. We then created a multi-ethnic beacon. That multi-ethnic beacon was actually pretty interesting. It's the one case you'll ever hear of me arguing against increased diversity. And the reason is if you have a multi-ethnic beacon, because of differences in allele frequency, 80 individuals who are CEU and, say, Japanese are more easily identifiable than 80 people who are all CEU, right? And that kind of stands to reason because of some differences and common variants between the ethnic groups. All right, this is one of the last slides, but unfortunately one of the more important ones. These are existing beacons that we launched attacks against. This is the 1,000 Genomes project that's got 1,000 genomes in it. The phase 3 of the project had 2,500 genomes in it. The biggest beacon's the Broad, that's got about 60,000 exomes in it. And one of the smallest beacons was a beacon of Native Americans and Egyptians, okay? And here the number of SNPs that we required for re-identification, we only need about 300 pings to this collection of 100 genomes to be able to figure out with 95% certainty whether somebody was in there or not. For the Broad, which was the biggest, we needed 200,000, but that's still less than your 23andMe data. Okay, so even if there's 60,000 people in the database with, you know, enough information coming off of an array, you are not anonymous, right? You can't hide among 60,000 people in this collection. And your children even can't hide among 60,000 collections. So it just gives you a sense of how easily identifiable folks are and why it's probably a bad idea to allow these anonymous beacons, even though most people want to use them for completely beneficent reasons, right? The issue is that it's still exposing a fair amount of information. We also were able to take an individual who we knew was part of the Personal Genome Project and was also in known variants, and we sent in 1,000 pings, and you could easily see that, you know, they matched known variants and they matched the PGP project. They were able to hide in this one, given that it's 1,000 variants and there's 7,200 people. We needed more. This was sort of the p-value that got returned. However, we clearly plucked them out from PGP. This Cafe Cardio, the reason there weren't any hits there is because it's a small panel gene. It's not a full genome ping. Okay. So with that, I just want to leave you with a couple thoughts. You know, this beacon problem is actually a big deal. You know, the Global Alliance has now modified their beacon protocol. They were allowing beacons of any size before. Now all the smaller beacons get put into a bigger beacon, hopefully moving away from anonymous pings, which I think is part of the big concern. The other is that people could choose not to give out the beacon metadata. Now, it's also possible to censor your beacon so it doesn't respond for very low variance, for singletons and doubletons. But of course, that's the whole purpose of a beacon, right? You want to find people that carry these rare variants that you may have also found in your clinical case. So, it's part of the decision making you need to make if you want to secure this genetic information. What can work? Well, we're now implementing better access control and thinking about a logging system because we think that's going to be really important as you try to distribute and make these data available to a broader community of clinical geneticists that want to use it. There's some limits to this work. In particular, we assumed independence among sites, which is a good approximation for some amount of pinging. You could do better with haplotype information. We didn't optimize, so you could actually get much better performance with smaller pings. We actually wanted to make the problem harder for us, but it is possible to do this. And an attack on a real beacon, if you're hiding the information, could be slow, might take a couple days, but if you're just throttling it through, it may actually be fast. All right. So with that, what we've recommended to the community is that we get rid of anonymous beacons, that for beacons under 1,000 individuals, you really limit the number of pings because of re-identification, that multi-population beacons unfortunately aren't as effective as you'd like them to be, so this is not a problem that you're going to solve simply by bringing all of this data together, and really the most important take-home is that we should get rid of this anonymous pinging because there's really no safe way of implementing that. With that, happy to take any questions you might have on any of the 3 vignettes that I discussed. Thank you.